for this kind invitation and introduction to my work. Today I would like to give you a broad and concise overview of our work on noroviruses in particular, so I will not speak on viruses in general. Noroviruses are still a human health challenge. They are a genus of the family of the Cultiviridae. They are simple, non-enveloped viruses, which means that they are not carrying glycans on their surfaces. And they contain a single-stranded RNA genome, which is surrounded by a capsid that is composed mainly of one major capsid protein, the VP1 protein. It's accompanied by a smaller protein, the VP2 protein, which has just stabilizing functions. Noroviruses, together with rotaviruses, are the most frequent causes of virally induced acute gastroenteritis worldwide, and this holds true for developing and developed countries. So the numbers given here may be not actual, but it is estimated that outpatients, there are 7.7 million in industrialized countries and 0.5 million hospitalization in developed countries, and at least 9 million hospitalizations in developing countries, with nearly 200,000 deaths per year worldwide. And among children, we have annually about 50,000 deaths. Noroviruses are a genus which comprises 6 genogroups, G1 to G6, and only the genogroups 1, 2, and 4 infect humans. The most predominant strain which caused pandemic infections is G24, and in recent years, in 2015, a new strain emerged, which is G217. A couple of other genogroups are infecting other animals, for example, pigs and mice and dogs. And the simple reason why these strains are more or less specific for their species is that we have no real functional animal model for studying human infection in an animal. The only animal which can serve as an animal model is oyster, because oyster expresses also blood group-like structures similar to the human structures, and we can use this model to some extent, but not for studying infection, but it's just the binding of the virus to the gastrointestinal tract. Well, as I said, the most prevalent strain currently still worldwide is the G2/4 strain, in particular the strain which has emerged first in Australia, in Sydney, in 2012. And during the year of 2015, a new strain emerged, G217, in particular in China and Japan. These two strains, which are currently the most relevant strains in human infection, show characteristic age distributions. G2/4 particular is infecting largely younger children and elderly people, whereas the G17 shows a more broader spreading infection pattern. So it is claimed that G217 has gained high affinity for carbohydrates, and this may explain its stronger spreading efficiency. Now, a virus to start or initiate an infection needs to bind to an epithelium, and the epithelium is covered by highly glycosylated glycoproteins. One typical example of these glycoproteins is the mucin MAC1 or mucin 1, which is inserted into lipid rafts of the epithelial cells and presents hundreds of oligosaccharides, O-linked oligosaccharides. And these oligosaccharides start with the addition of a GalNAc to the amino acids serine and threonine, and these galactosamine residues can then be extended to a core 1 structure by addition of a galactose, and this core 1 structure then can be further elongated either by addition of sialic acid, which serves as a stop signal, so these structures are dead-end products which cannot be elongated anymore, or a branch is introduced by introduction of a glucanac residue at the position 6 of the GalNAc, and this glucanac residue is then extended to form so-called polylactosamine type structures, which can carry at their ends blood loop structures, which contain fucose and GalNAc and galactose. So these structures are also found on those mucins which are secreted. So in the gastrointestine, also in the stomach, we are confronted with these membrane-bound mucins, which are the target sites for the virus. And we see also secreted mucins which form viscoelastic gels on the surface of the epithelia and prevent a direct contact of viruses and bacteria, of course. So they also express the same structures principally, although each mucin which is secreted, for example, in the stomach, has an organ characteristic glycosylation. So one of the mucins which is expressed in the stomach is the MAC5AC, and another one is secreted by deep gastric glands. This is the MAC6, which shows a very characteristic distinct glycosylation. This slide I show just to give you an overview over the structures which are relevant in the context of this presentation, derived from either type 1 structures, which is galactose linked to subterminal glucosamine, or type 2 structures where the linkage is 4 to the glucosamine. The blood group structures are derived from by either the secreted gene-dependent fucosyl transferase, which introduces fucose to the 2 position of the galactose, or by the Lewis-specific enzyme, which introduces the fucose to the subterminal glucosamine. The structures which are relevant in the context of milk oligosaccharides are shown more or less on the left-hand side, and this is why I asked Lars Bode whether he has any evidence on the formation of these type 2 structures, which can also be formed. Let me go on. Now we are searching for anti-infectiva based on natural compounds, and the natural compounds we first thought of are human milk oligosaccharides, and we asked the question whether these can be effective competitors of viral adhesion to epithelia. Now, the idea is quite simple. The virus is a multifunctional or multivalent lectin. So on its surface, it expresses multiple copies of the VP1 protein, and the protruding domains of these VP1 protein are forming a lectin-like domain, which is able to bind carbohydrates. And also on the other side, on the epithelium, we have a multifunctional glycoprotein. So this interaction to overcome will be a challenge for monovalent short-chain carbohydrates, but we thought we should have a look at the larger structures expressing more than one fucose, and to use these larger structures as potential decoys to inhibit this binding process. The VP1 protein, the structure is shown here on the right-hand side, is composed of 3 domains. The S domain, which is a structural core domain, and this is additionally by 2 further domains, the P1 and the P2 protruding domains, which form at the end this carbohydrate binding pocket. We're using this VP1 protein, which forms spontaneously to virus-like particles, and on the other hand, a dimer of the P1, P2 domains. This dimer was used in crystallography studies and co-crystallography studies with milk oligosaccharides, whereas the virus-like particle was used exclusively in solid phase binding studies. The co-crystallization was performed with a series of milk oligosaccharides, either defined standard compounds or mixtures of undefined oligosaccharides, because our initial idea was to see whether complex oligosaccharides exceeding the size of decasaccharides, for example, could bind to the lectin of the P domain and could in this way in the crystal be entrapped. And these crystals could then be analyzed by either X-ray analysis or by multi-mass spectrometry. So we could have been able to identify those structures which were preferentially entrapped in these co-crystals. Unfortunately, these larger structures never gave crystals, so we had to give up this project, and we could demonstrate for a couple of viral strains, in this case here for G210, the Vietnam strain, that only smaller sized oligosaccharides could be co-crystallized. And here, for example, 2-fucosylactose and 3-fucosylactose, although they have structurally nothing to do with each other, were able to crystallize within the pocket. On the other hand, other structures which have a structural relationship to the 2-fucosylactose, like the lacto-N neo-fucopentaose 1, did not co-crystallize. The genogroup G24 has evolved within 40 years. So in 1974, the Washington species, the Washington strain, which is shown here, this strain shows a strong binding to blood group A structures, whereas the later evolved Sydney strain from 2012 shows a diminished binding to blood group A, which means that within these 40 years of evolution, the strain has already changed dramatically. And here you see the many mutational changes which have occurred and which affect in some way the structural aspects of the binding pocket of the VP1 protein. And this results finally in the orientation or in the different orientation of this loop-like structure here, in particular, and to different binding of the 2-fucosylactose shown here as an example. So already these few mutational changes here occurring during 40 years have dramatically changed the specificity of the binding groove. So the result of this mutational change is that the newly emerged strain from 2012 binds much better to blood group H structures and less to blood group A structures. Also, the newly emerged strain G217 shows an important mutation when compared with the first emergence in 2002 in Saitama in Japan. And this mutation refers to the replacement of a valine at 444 to a tyrosine. Lectin pockets are generally characterized by the phenomenon that they contain very often aromatic amino acid side chains, which are highly conserved. And we can speculate that this mutation here has made this binding pocket much more sugar-philic. Now, we in Cologne were interested in finding structures with a higher binding potency to the virus-like particle in the fraction of high-mass milk oligosaccharides, which were kindly provided by Clemens Kunz from Gießen. He provided us 42 fractions of high-mass glycans, which were characterized by 2 to 6 fucose residues per chain. Well, our strategy was based on an array which is composed of neoglycolipids derived from oligosaccharides. These oligosaccharides have reducing termini, and we can couple to these termini phosphatidyletherdonamine, and these neoglycolipids are then embedded into a lipid layer and expose their carbohydrate chain to the aqueous phase. So we simulate in this fashion the situation on an epithelial surface to some extent. And in this model, which was developed by Tanfasi in the late 80s, we have initially tried to show that we can use this approach. In this case, we used the less relevant strain G210, which is more important in oyster infection. And we could demonstrate that one fraction of this complex array was most potent, and this is a fraction of milk oligosaccharides here, of large, high-mass milk oligosaccharides. Now, we did the same with the 42 fractions provided by Clemens Kunz, and we could demonstrate that not all fractions were active. Most fractions were inactive at all, but a couple showed some significant binding activity. Now, to get insight into the structural requirements in case of the G2/4 binding, we use also a couple of standard compounds like lactofucopentaose 1 and the neo isoform, and lactofucopentaose 2, the Lewis A expressing pentaose. And this clearly showed that the virus-like particles could bind to both blood group H structures, but not to the Lewis A structure. So there is some degree of specificity in this binding. But in contradiction to other reports from other investigators, there is no discrimination between H1 and H2. There were reports from researchers working with saturation transfer difference, NMR, who could show that there is a strict discrimination of the virus particles between blood group H1 and H2, and also data from X-ray analyzers which said the same. But actually in this maybe more realistic experiment, these tiny differences in affinities were not or did not become visible at all. So the virus-like particles does not discriminate between these structures. Now, we isolated the most potent, most binding active subfractions of some of these fractions, and did mass spectrometry analysis, and could demonstrate that these highly active fractions were characterized by multiple fucosylation. Up to 4 fucoses were here found in this deca saccharide or nonasaccharide, and we could also gain structural information with concern to the type 1 and type 2 structures at the terminus of this branched structure. Now, what we saw on the level of the neoglycolipid arrays, we could reproduce with mucins isolated from gastric juice of individuals which had been defined with respect to their Lewis and secretor blood group status. And we could show that the virus-like particles bound preferentially to the Lewis B structure and to in case of the Lewis negative material, to blood group H structures, but not or much less to Lewis A. Now we tried to inhibit the binding of these virus-like particles with a series of milk oligosaccharides. And we started with a 2-fucosyllactose, which can be referred to as a reference, and with lactofucopentaose 1. Well, both showed inhibition at high concentration. So in case of the lactofucopentaose 1, which is actually or should be theoretically one of the best binders, we found IC50 values in the range of 50 millimolar or even more. We now have better data which are not presented here, and I can tell you that all higher mass oligosaccharides like lactofucopentaose 1 and the respective neo isoform both have these lousy affinities, obviously, in this system. 2-fucosylactose and 3-fucosylactose have IC50 values in the range of 15 to 25 millimolar. So obviously, these monovalent oligosaccharides are less potent in the blocking of viral binding, and this could be due to the valency of the lectin on the one side and on the valency of the oligosaccharides in case of the epithelial surface. So the question is, how can we compete with these multivalent components in this system? And this is an artificial approach, so to say. We thought we use a scaffold like alpha-cyclodextrin and couple to this cyclodextrin fucosyl residues simply by an acid reversion technology. And the fucose was shown by methylation analysis by GCMS to be linked exclusively to the 6th position of the glucose in this ring structure. And the fucose was linked alpha-glycosylically mostly on the basis of NMR analysis. So we could isolate at the end mono to hexa-substituted derivatives and could test these as binding inhibitors, and we could see that with the increased degree of substitution, there was a tremendous increase in efficiency of blocking. So obviously the valency is the most important point, and you will agree with me that these structures shown here have nothing to do with the blood group structures in milk or on an epithelium. So it's just the fucose which is important, and this fucose should be presented in a multivalent fashion. And this led us to the question whether there could be other compounds in nature which could substitute for these blood group specific structures in milk. And we found these fucans or fucoidins, which are expressed in many marine invertebrates and in the cell walls of many brown algae, in particular in the fucalis and the laminarialis. These fucoidins are characterized by long chains where the core can be either completely composed of fucose only, or it can be repeating fucose and galactose, either 1,3 or 1,4 linked. Here is a structural model of the Fucus vesiculosus fucoidin, which is characterized by a high degree of sulfation. So every second fucose of this structure is sulfated. So these fucoidins are known for some time, and they have been shown to exert a multitude of bioactivities, among others anticoagulant and antithrombotic activity, anti-tumor and immunomodulatory activity, and so on. and among others also antiviral activity, for example in the poliovirus, adenovirus, and against HIV and cytomegalovirus and herpes simplex viruses. But I have to say all these effects have nothing to do with the binding competition, so the mechanism here is completely different, and it's totally dependent on sulfation of the fucoiden. Now we have tested various forms of fucoiden as a competitor of the viral binding to gastric mucins, and we could demonstrate that it is highly effective as a competitor either in the native form or in a desulfated and partially fragmented form. So you can see here the masses of a partially fragmented form which has been generated by solvolysis. And here you see a partial structure of those oligosaccharides shown here, which were highly active in the system. Well, interestingly, when we tried to entrap the oligosaccharides generated from fucoiden in a co-crystal with the P domains of the virus, we could demonstrate that only fucose shows up in the binding pocket. So this is exactly what we had expected. There is a strong effect, but this effect has nothing to do with the specificity. It's just the fucose which is linked. So it's a low affinity binding, but with a high avidity. This slide just shows the concentration dependency of the effect, which should be seen in a competitive system. And this slide may be interesting because it demonstrates that the viral particles are able to survive in some way even at lower pH conditions as they are given in the stomach. So down to pH 2.5, they are quite active, actively binding to gastric mucins. Currently, we are testing a variety of different preparations of fucoiden from different species, of course, which is not shown here on this slide. And we are comparing the pure stuff with the more purified stuff, because purification means that solvents have to be used, that oxidation processes are involved in all these things, and all these chemical treatments have influence on the activity. Now, this is one of my last slides. We are currently trying to establish oyster as a model for binding or accumulation of virus particles in the gastrointestinal tract of these animals. We could demonstrate that actually the strains G11, G24 are accumulating in this system, but interestingly not the G217 Kawasaki strain, although this has been claimed to have a higher affinity to blood group-like structures. But obviously the blood group-like structures recognized by this strain are not expressed in oyster. We have already initial data in this model and can tell you that there is a certain degree of inhibition exerted by 2-fucosylactose, the milk oligosaccharide, in this system. So principally, this model can work. Let me sum up what we have done. We have shown that the binding of GT4 VLPs to human gastric mucins is sugar dependent. The most important sugar in this event is the fucose in blood group H1 and Lewis B-related human milk oligosaccharides. However, the affinity of this specific interaction, or at least the apparent affinity, is quite low in this system because of the multivalency of the interactors. The IC50 values are in the range of 20 to 60 millimolar for these tested milk oligosaccharides. We have demonstrated the valency, the importance of the valency effect in an artificial fucose dendrimer with these compounds, and have demonstrated that polyfucoses like fucoiden and fucoiden-derived oligofucoses are potent inhibitors of viral binding. The sulfation of fucoiden does not play any role, so the mechanism of this inhibition is completely different from other viral inhibition. Okay, I thank you, and I just would like to mention my collaborators, Horst Schoten and Wassily Morozov from Mannheim, who are doing these oyster experiments, Bernd Hansmann, who is responsible for the X-ray co-crystallography, Stefan Jennewein, who provided a couple of milk oligosaccharides. Tenfasi was involved in the initial neoglycolipid array, Clemens Kunz for providing the milk oligosaccharides of high mass, and Dolores Diaz from the University of Cologne for performance of NMR work. Thank you for your attention.
Thank you very much. This paper is now open for discussion. Yes, Arnel?
Thank you very much for the presentation. I remember a paper that must be 15 years ago or so, where they studied the inhibitors in human milk of binding on norovirus, and found that that was not the soluble oligosaccharides. The most inhibitory factors were actually two glycoproteins, and what they're having, that was mucins and bile salt stimulated lipase. And what they have in common is the tandem repeats that are O-glycosylated.
Yeah.
Do you have a comment on that?
Yeah, of course. Well, this is exactly in line with what I said before. So the glycoproteins, in particular the mucins, are the much better competitors because they are multivalent. And in particular, MAC1, which is one of the most prevalent mucins in human milk, on the milk globule membrane, it's the most important glycoprotein. It's also secreted, and it's also shed from the membrane, and you find it in high amounts in milk, also in the whey of the milk. And we have done a lot of structural work in the late 80s on this mucin. And actually, this mucin would be the better player in this system, but not the monovalent oligosaccharides.
Well, I was actually thinking, is it the fact that they have tandem repeats that are O-glycosylated, so you have many different binding sites on the same protein? Do you think that's the key?
MAC1 has, for example, up to 120 repeats. One repeat is a 20-meric peptide, and this peptide contains 5 theoretical O-glycosylation positions. They are not all substituted, but most of them. So you can end up with hundreds of oligosaccharides, very complex, very heterogeneous, of course, but among these, also these blood group-like structures.
Thank you.
Other questions? Dr. Bode?
Just did a quick calculation. You said it was 20 to 50 millimolar, right? So that would be 10 to 15 gram per liter of oligosaccharides, which is exactly what we have in human milk. So total human milk oligosaccharides, though, right?
Total, yes.
Right. So if we think of human milk not just being one individual component, but having glycoproteins, having glycolipids, having some antibodies, having all kinds of other defense mechanisms, I would argue that it's not one particular compound that has these effects, but the interplay of all of them together. So if you add these oligosaccharides, and maybe it's only in the 10 millimolar range that it's effective, or 5 millimolar range. So we're far away from the IC50, but together with all the other components, it might actually make sense. Right.
But actually, we have no animal model, and we cannot test all these compounds, and so we have just these artificial systems, and we need to stick to these.
Sure. Yeah. But I think it's a good argument that milk is not just the sum of different components. It's an orchestra really of of multiple different defense mechanisms that play together in the end.
But I'm not arguing totally against the idea that milk oligosaccharides could make the race. So, for example, 2-fucosylactose is tolerated by an adult person up to 20 grams per day. This is enough. Yeah.
More questions to Dr. Hanisch? Yes, Dr. Ogra.
I really enjoyed your presentation. I just was wondering what is so unique about the oyster in the context of norovirus? And do you see norovirus infection in any sea animals also?
No, so far not. Well, the oyster has one advantage. We know that those viruses or noroviruses which are affecting or accumulating in oyster are generally also infectious in humans. So this is known for a long time. So oyster came initially into our mind because this is a known trait. Whereas pig, for example, have other strains. So they have the G3 strains, and dogs have other strains. So can't use any of these because they are not infectious in humans.
Well, I was wondering, do you see norovirus outbreaks on cruise ships, for example,
Yeah.
a lot.
Yeah.
So I was wondering, is there any particular relationship to the...
It could be. I agree.
Any other questions? If not, I suggest we take a coffee break. We were up admirably early. Natalia has an announcement before we go and have coffee.